final signal for the cell, changing the DH ratio. And when we are using deuterium depleted water, even that system is activated, but the cells are not able to lift up the DH ratio to the threshold, or it takes much longer time. And this is the reason that we can finally disturb the whole metabolism and no signal and no cell division, and finally we can trigger the apoptosis. There was another experiment which partially can prove that, and Gábor Laskay will show another one. In that experiment, we made an acid load. So we brought down the pH in the cell to about 3, and then we checked the eminently sensitive sodium hydrogen antiport, how can it work and how can recover the pH in the cell. And as you see, the curve is different depending on the deconcentration. So we can suggest that somehow, for example, the sodium hydrogen antiport is sensitive for the deconcentration, and that can modify the activity of that transport system. I have shown you that in vitro growth, the lower was the deconcentration, the bigger was the inhibitory effect. But the real question was whether we can modify the expression of COX-2 gene. It is well known that the COX-2 gene has a key role in tumor development. It is responsible for enzyme which synthesize prostaglandin. And what we could see here, we could see a clear correlation between the deuterium concentration and the expression of COX-2 gene. And at the same time, we found correlation with the prostaglandin concentration and the deconcentration. The lower was the deconcentration, the lower was the lower was the deuterium concentration, the lower was the concentrated prostaglandin. And the question was whether this prostaglandin has any role in this inhibitory effect. So we made another experiment that we add back the prostaglandin from outside, and we checked whether we can diminish the inhibitory effect of deuterium, and we could do that, because as we increased the prostaglandin concentration, simply disappeared the inhibitory effect of deuterium depleted water. So that could be one example that the deuterium depleted water can intervene into that signal system and can modify the enzymes and gene expression which are closely related to cell growth. In another experiment, we just checked the MAP kinase, which is labeled within 2 minutes when the cell got a signal, and you could see it didn't happen when we used deuterium depleted water, suggesting again we can modify this system. Later we went on and we chose the DNA chip containing all the 30,000 human genes, and this is the results what we got. It was what we found at the first sight, that a couple of hundred gene expression were changed in deuterium depleted water. So that answer was so complex that we decided not to go and not to continue in that direction. We chose another DNA chip with only 500 different genes. These were kinases. And repeating several times and choosing the real good responding genes, we finally could say that 7 genes responded extremely well to deuterium depletion. There was an up regulation and there was a down regulation. Combining all these results and all the results of the experiments, we could say that there's an extremely complex response to deuterium depletion, and I think there are a lot of work to do and to discover all the background of this process. So the conclusion, we say that the nature occurring deuterium may have a key role in cell cycle regulation. The changing DH ratio can be the signal to introduce to the induction of numerous cell process. The changing DH ratio can simultaneously modify the expression of genes and the activity of certain enzymes. We say that the results show that it had unknown summer regulatory systems may exist in the cell, And this deuterium-hydrogen submolecular laboratory system can be a new target for anti-cancer drug development. Before I finish my talk, I would like to ask you to think about, if we take only one animal cells, we can see how complex its structure. We know that within one human cell, there is a DNA with a 1.8 meter length. It contains 3,200,000,000 base pairs, and all this DNA, for example, is packaged in a small cell, which size is only 30-40 micrometer. We have discovered the metabolic pathways. It's extremely complex. The question is how the life can organize that extremely complex system. And I say that should be the submolecular regulatory system. And I think if we can proceed, we can make a big leap. So we used a computer and with a huge piece of equipment, and it could make only a couple of calculations, and that chip can do maybe 1,000 times, million times more than these big, very old computers. So I say that the life can organize that very, very sophisticated system, because on a submolecular regulatory system, they can simultaneously organize the gene expression and proteins. And that way, somehow, they can harmonize all the processes in the cell. That's all. Thank you for your attention.
Thank you for your presentation. The paper is open now for discussions. Any questions or comments, please?